Yeah. Hi, Dave. Scott's typing. Boy, are you typing.
I'm saying howdy to our live chatterers.
Yeah, but you're coming in like thunder. I think your mic settings are high or something, but you're really coming in like thunder. It's like boom, 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 boom. Yeah. Anyway. Oh, 2 viewers. We 3 viewers. KCA Randy, we got Dave, we got Laborious Cretan. That's pretty good. Anyway, why have we gathered here today? Now, creationist nonsense appears because somebody puts it out, in a video or somewhere online, and they come up with some devastating thing for evolution that becomes a fact, and they put it out there, and they're wrong. They misread the paper, and they're wrong, and it will never go away. And when you confront them with it, it still won't go away. You can confront them with, you can tell them, look at this thing that's happening right here in front of you. And they'll say, oh, that's not happening. It's just like, what in the hell? Oh, KCA Randy, life is good. Life is good. Anyway, so such a thing happened on Rebekah's channel when she was interviewing Uditha Jayatunga, who wrote a book. And this guy is, he's a physical therapy doctor. He works on people's, who can't walk, who have disabilities. And he is really thick. I mean, some of the stuff he says in this video is just astounding. And he wrote a book, of course, on creation. So anyway, it's just like, shit. So I guess the best place to start would be here with the part I had a problem with. Okay, this is him stating a factoid that is going to become a creationist point in the future. We're going to hear this again. It's never going to fucking go away. It's going to keep happening, and that's kind of what I want to show in this video. So, and we'll get into a little sciencey stuff along the way, if that's okay. So let's see what he says. Let's see if I can play it. I hope I got the right part.
This is the specific study saying that the latest and largest genetic association study of intelligence has identified 206 genomic loci and implicated 1,041 genes, adding 191 novel loci and 963 novel genes. So novel genes, these are new to humans. These are not seen in other animals. How can such a massive number of genes suddenly appear in humans?
Okay, 91
novel loci and 963 novel genes. So novel genes, these are new to humans. These are...
Okay, that's it right there. That there are 1,000 genes that affect the brain that are not in any other animal. They're only in humans. They're orphans, or orphan genes, you'd call them, as they're completely novel. So that's what he got out of this this study and this paper. And I went and I looked at the papers, basically, that the one he's identifying here is by Goryanova, I think it is. We could go ahead and take a look at the papers, I guess, first. Am I looking at Uditha? Okay. I don't have these labeled, so I got to cycle through them. Okay. So Goryanova 2019, Gene Cells and Brain Areas of Intelligence. So what these guys are trying to do is they're trying to find mutations among humans that affect intelligence, and they're trying to show different changes in alleles across humans will have an effect on whether one person is smarter than another person. And also they're looking at dementia and Alzheimer's and a few other diseases of the brain. So they're trying to find a correlation, right? And so Goryanova wrote a review article that reviews research on this matter and identified 206 genomic loci, implicated 1,041 genes, adding 195 novel loci and 963 novel genes. You see how he says novel, right? Well, then to previously associated with, from Savage et al. 2018. So I went down to, I'm not showing my screen. Shit, Scott. Oh, okay. No, that's not right either.
Sorry, I was capturing a link from Dr. Joel Duff's channel.
Okay.
Just like you are heading off a creationist talking point that won't go away because it's already in the ether, in the creationist zone of brain dead. He recently, like on December 30th, did one titled "Preserved Cells in the Devonian Fish: A Hemoglobin Challenge Preview." So, in other words, he's heading off claims that people are going to make about finding tissue in Devonian fish fossils.
Okay. So, anyway, I was reading the paper, the part I have highlighted here. I'm hoping people can read it. Where he says novel, Goryanova says novel, and then he refers to the paper by Savage et al. 2018, right? And so we go look at Savage et al. 2018, and he says 930 new genes right here, not novel, new genes. And if he had said novel, he still would have meant new, because I read the papers. Not only did I read the papers, but I threw them into Notebook LM and I asked questions about the papers to make sure I didn't get it wrong. I went through and I talked to ChatGPT about it and tried to come at it from a number of different angles, and it said no. So what this study is about is you take, I think he took 269,000 genomes and found basically SNPs, or what the hell, nucleotide polymorphism, single nucleotide polymorphisms in 906 loci, right, or 205 is genomic loci. And then he correlated that with intelligence tests and with some diseases and a bunch of other stuff. And so that's what he said. So what do we have here? So it says identifying, let's see, 939, okay. 1,016 genes, of which 939 are new to this study. So it doesn't say anything about novel genes. It doesn't say anything about orphan genes. If you search these two papers, and I've linked them in my chat, my description, you will find nothing that says anything about primates or other animals. They're talking strictly about human beings and about variations within humans. They never said that there were 1,000 genes that humans have that chimps don't have. That was entirely something that Uditha read into this whole thing just by snipping that one little piece, which is ridiculous. And
so do you think he went and did a search for novel genes or new genes?
Yes.
Found that sentence and said, aha, this proves my point.
Yeah, and he never bothered to go and read the paper, the other paper, basically, that, let me see here. Okay, that one. He never bothered to go and read the Savage paper to see that it didn't say novel, it said new, and it wasn't about finding novel genes. He just assumed that he had something. It's like, oh, evolutionist, I got you now.
So,
anyway,
is it possible that he just got his papers mixed up and that there's some other paper out there making that claim? Has he had time to look and verify and say, oh, I'm sorry, I quoted the wrong paper?
They haven't got back to me.
Okay.
I did this 5 days ago, and Rebekah hasn't even gotten back to me. I'll show you this in comments.
Okay.
So here's a comment I made. If there's some serious misinformation, you need to correct it. Let's see. So I mentioned Goryonova's paper gets a number from another paper, the Gloss study, Savage, and I mentioned that. Humans, they are not, okay, humans, these are not seen in other animals. How can such a massive number of genes, okay, this is his quote, massive number of genes suddenly appear in humans. And I said the Gloss study added 900 genes to the study. They didn't say there were 900 genes added to humans. Nowhere in either paper do they talk about primates or other animals and find for new human genes. Please at least browse the paper before saying stuff like that. Thank you for your attention to this matter, MBB. I was being polite there. Right. Okay. So then Micah comes back and said they do say they are novel genes. And I just told them, no, they don't. They say they're new genes added to the study in the paper. But he's doubling down here. Okay. So 963 novel genes to previously associated with cognitive ability or something. I don't know. Anyway, I said in the Savage study, they say new genes. Gorionova changed it to novel for some reason. In any event, there's nothing anywhere that will tell you we have 1,000 genes that other primates do not. Pick a gene, give it to me, and I will find it in the chimp. Okay, so then this guy is pretty cool. He's been very polite with me. He's been very, very reasonable. Frank Hubini 958. The author's manuscript says novel rather than new, and he says I find it puzzling why the number. Okay, you seem to assume that there are no novel genes in humans since you claim they're all in chimps. Actually, I think we found like 74 of them or something like that in humans, but we'll get into that. That's orphan genes that we're talking about, right? How do you explain the functional differences between chimps and humans without them? Well, it's regulatory changes, small changes to regulatory regions, and some other goings on that affect development. That's why we have a bigger cortex. Anyway, and then he says, from my perspective, human genes are all novel genes because there is no evolutionary connection between animals and human beings. Some genes might have been reused by the creator as designs, but they did not reach their position in human beings through evolution. So we know where he's coming from. And I said, I do not know what paper you're looking at, but here it is again. The difference between humans and chimp brains is similar to the different Savage found within human genome variants. So basically, what changed between the chimp and humans when we split for 12 million years are these regulatory regions that change how these genes express and when they express, right? So that's the point I'm making. Let's see. And then Misha comes back and said, why are you trying to split hairs? Is it because you cannot face the science? It's like, I can't face the science? I read the fucking papers. It's like, read the papers and see what they're saying, for God's sake. It means they have not been identified before in humans or any animal. It says 963 novel genes, not been identified before. They've been identified forever. Are you suggesting that all the genes we have are found in chimps? If so, why are we different? Are you also saying cognitive differences we have chimps are not associated with any new genes? Yes, I'm sure they have found a few new genes, a handful that turned on all of a sudden, basically, or were promoted where they didn't think they were promoted in other primates. We've got around, okay, and then he goes, we have got around 15% difference in DNA per the latest data. So he's going to reiterate that fucking nonsense that we have a 15% difference with chimps.
So just to make sure I have the issue well in hand, they quoted a paper that used the phrase novel genes, 963 novel genes. That was from a paper, or to support that, they cited a paper, the one that you showed us, that didn't say anything about chimps or novel genes. They just added genes to a study that had previously not had those genes in the study.
Right, for cognitive correlation with IQ.
Yeah,
that's what they're doing. Right.
So did they not even go to the source paper? They just are dying with the guy that said novel genes instead of new genes added to the study?
So, could you repeat that? I was reading something.
Now remember, I'm assuming that these creationists cited a paper that used the term novel genes.
Yes, Goryanova.
That paper cited the one that had added 963 new genes to a study. It didn't say anything about novel genes or anything about chimps.
Right.
So did they just do that first paper? Did they just like read that first paper and just die on the hill of this paper says novel genes, therefore it's novel genes and you're wrong? Is that what
Goryanova? Yeah, right. Yeah, that's but Goryanova didn't intend to do that. Goryanova was just switching the word novel with new.
Right, but you're seeing how the creationists are misinterpreting it. And but they didn't go to the source of the source. They didn't go to the original source that they quote to confirm that that's what was being said.
Yep.
You see, this is one thing for everybody to learn here. Let's take the creationist misinformation project out of the picture. Scientists screw up. If you read a paper that says something that you want to pass on and quote, you better go to their source to make sure that they're reading the papers that they're citing correctly. Scientists are wrong and can be wrong. Scientists probably mess up all the time, which is why you have to go and do a source analysis. I'm thinking of our friend who wrote The Rocks Were There. Jackson Wheat and his partner, AJ?
James Downard,
James Downard, Downard. So, always go the extra mile. Don't take shortcuts in trying to pass on scientific information.
Right. So then Misha 2704, the rocket scientist, he said, the sentences mentioned are directly from the paper as published. Do not try to twist it just because you cannot face it. And so I went out and did some work.
Somebody is letting some ego really get in the way.
Right.
And you weren't even rude in that paper. You just said you need to correct this.
I know. I was being really nice. Anyway, so I went out and did some work on my genome. I'd got my genome project with all my data that I downloaded. I read the entire list of new genes for human and chimp. Genes found only 28. Well, actually, I found 23 because I made a mistake in my software. These lowercase ORFs basically were not finding something because I forgot to switch it to uppercase and whatever. So these are the genes that I found that actually you can find in the human, but you can't find it in chimp. Okay. In my database. That's very important. And let's see. And then, but interestingly, I found 3 genes that they use in their human study that I could find in the chimp, but not in the human. So there's a reason for that. And the reason is synonym problems and annotation issues. Sometimes these genes have different names, particularly when you see something like CCDC. I think that might be a coding region. And I don't know what the other one is, but anyway, so that's interesting. I mean, why do you find them in the chimp but not in the human on my database? I have to look into that, right? Because I got a problem. Anyway, so Examining Origin says, hi, Mike, thanks for your substantive comment. If there's a serious misinformation in the video, I definitely want to correct it. Your comment is difficult to decipher. Please say, I don't think it was that hard to figure out. Please take some time to clearly state what the problem is. And I said, the study by Savage.
That's what you're dealing with there, the inability to comprehend.
Yep. The study by Savage was for 270,000 individual humans. I think it might have been more. Previous studies looked for variants within humans that correlated with IQ and disease variation in humans, but they used 24 genomic loci. The Savage study simply included more genes in their research, 205 instead of 24. This was misinterpreted by you and your guests to mean that there are genes in the human that have no homologs in other species of primates. The reference papers made no mention at all of other animals. Nowhere in those papers did they claim that there are about 1,000 novel genes in humans. And Misha says, once again, it says 963 novel genes. What else do you understand from this statement? I've been trying to explain it to him.
You need to tell him he's gone from dying on the wrong hill to being ground into dog food on the wrong hill, because he's not going to the source paper and realizing that the person who said novel genes is misinterpreting it.
Yeah. These two papers, very hard to read. I'm pretty sure that they didn't come anywhere near reading those papers. A lot of neuroscience, a lot of statistics. Okay, the claim that these novel genes are unique to humans, and this is what I'm writing to her to clarify, are unique to humans and not found in other primates, a misunderstanding of the scientific terminology used in these texts. The sources demonstrate that the term new and novel refer to newly identified statistical associations in the scientific literature, not biological genes that spontaneously appeared in humans. How much fucking clearer can I get? Anyway, so then Frank Hubany, 958, responded, and he agrees with Misha, whatever, and goes on and says again, this. Third, the question seems to be related to the definition of novel gene. Did they say, okay, now I found a definition of research. Yeah. Novel genes based on phylogeny or whatever. He's got a link, and that'll tell you about orphan genes, right? But no, that's not the context in which they use the term. Novel can mean new too. We say novel all the time in our everyday conversation.
That's what novel means. New.
Yes. However, isn't that what the term novel gene means? Or wouldn't novel gene and new gene be synonymous? Would they? I don't know. Not if you're saying I use new genes in my study, it wouldn't be synonymous. Anyway, and
they didn't sort through and find a list of 963 orphan genes and then put them in the study. They were just genes that they put into the study. Now, is there a way to identify or as novel gene in the genetic sense, the geneticist sense, and search through that list of 963 genes and say, this is not a novel gene, this is not a novel gene, this is not a novel gene? And
we'll get there.
And say, okay, all right.
We're gonna get there. Okay, I do not know how to make this clear. I mean, you understand it. I'm sure everybody listening understands it. Why don't they understand what I'm saying? They're not hearing the words coming out of my mouth. It's like, I just, nothing to do with orphan genes. And you need to understand the paper, not the dictionary's idea about the word. I downloaded the supplements. Okay, so I got the supplementary data. I have a program that pulls a gene for both human and chimp. The supplemental data show 939 new genes and no data for the previous study. And then what do we have here? I believe there are 74 genes that we have found in humans that are considered novel or orphan genes. But that's another topic that we can say a little bit more about later. Anyway, I have the most recent T2T telomere to telomere data from NCBI, and that's the data I'm basing this on. And then he complimented me, and he wants to know how I did what I did. And I told him, I put the entire list into a Python array, blah, blah, blah. I told him, you're not going to like it. You're not going to want to do the work that I did on this, basically. So let's see, what do we got? We can look at, let's see. Oh my goodness. We got that. We got that. What did I miss? What did I miss? We got that. Oh no. I'm missing a page. Oh, anyway, let's see if I can pull this up. Hang on a second here. Gorianova, PDF, numbers, okay. Coming up. I hope I don't kill my computer. Okay. So here is the supplemental data for the paper. And let me see here if I can look at this. Intelligence, ADHD. So I think it's S24. S24, pleasant study. Maybe it's S23. Overlap. Oh boy. Where are they? Okay, here we are. Okay. So I'm going to have to make this bigger, Anna. Because you can't see that. So we'll go to 150. See if you can see it. Can you see it now? Kind of? Okay.
Kind of. It's still small.
Okay. So he has a list of all of his 1,064 genes. So he has gene name, he has evidence from the present study, and then he has previous source, okay? You see that previous source is blank for a number of genes. So ADTRP. So what I did is I took out all of the genes that are blank for a previous source. So these are the new genes he's talking about. He added these genes to the study. And I went through them all and came up with, let's see, where's Jupiter? Where are you, Jupiter? Jupiter. I don't understand why this isn't happening. What's happening here? Oh, let's see. Let me go look at Jupiter. I don't know. See if I can find this. Sorry about this. I had everything all set up so cool. And oh, here we go, Jupiter Lab. Oh, there we go. Okay. So, yeah, that's something else I did. So I tried a few genes at first, and then I went ahead and I wrote this compare gene presence function, basically that goes out to my databases. I have two databases, one for the chimp, one for the human. And I run this compare gene presence, and I come up with found in human 860, found in chimp 840. So here are the 23 genes that I could not find in the chimp out of that 1,000 or 939 genes that I checked. Okay. So I took all the new genes and I went and I found it. I found them in the chimp and I found them in the human. What else can I tell you? Except for these, right? And so then I started looking at a few of these, and every one I looked at, let's see, like these OR ones, I think those are open reading frames. Those are frequently different between chimps and humans. So you're not going to find, like on a chimp, it'll be annotated as a loc 9562753. And on a human, they'll have another name for it, right? And then I found a few that were only in the chimp. And then it goes through and it does all of that. So I went and looked at PCDH1, which is a protocadherin, which we can talk about a little bit. Very important in development of an organism. These cadherins hook two cells together, and they can make it so that cells can move and migrate, or they get sticky and they end up in a certain brain region, right? So that's a really good candidate for something that would affect our intelligence or affect the difference between humans and primates. So I decided to look into protocadherins a little bit. And then up here, what did I do? Where did I do it? Oh no, that's build summary, no. Okay, build summary. Go back up here. You're thumping.
Oh, sorry, I'm just saying hi to Bree.
Bree, I saw Bree. Hi, Bree. Direct silencer with CDS. I don't want to do that. Why did I go way down here, Scott? Oh, here. Okay. So this is PCDH1, which I found in the human. Oh, you can't see it. Okay. This is a picture of the human PCDH1, not AH1, just H1. And this is the human, and this is the chimp. And you can see how similar they are. They're in slightly different positions because of retrotransposons and other mutations that happen. But you can see that they have the same exons, right? And then I went down and did the same for PCDH, and it's not showing PCD. It wasn't showing that. Okay, D gene, PCDH1, or print PCD. Oh, no, up here. Okay. So I only found it in human. I didn't find it in chimp. And this is the other one, PCDH1. So then I went ahead and I found out, because I had the gene information, I downloaded the entire gene from the human, PCDH1. And what did I do? What did I do next? I ran a blast on it. And here's my blast. Okay. So I blasted it against the chimpanzee, right?
You know, there's a lot of people I want to blast these days.
Right.
But that's totally different.
Yeah. So I have my gene information here, and what this gives me, these two things are the beginning of the gene, and this is the end of the gene right here. And then I went ahead and blasted it, and I found human 5 is chimp chromosome 4. And I found it in, I believe, in chromosome 4, right here, chromosome 4. And you can see these vertical lines mean an exact nucleotide match. You see these gaps here, that means that there was a gene, there was a nucleotide. Oh no, the gap is the difference. A was converted to a G. So this is that whole section, even though they don't have it in the chimp annotated as PCDH1, I still found it. I found a match for 57,925, including the introns, the exons, the lines, the signs. And you can see that there are some differences here, right? There are some deletions and insertions, but very, very light, like probably amounting to about 0.05% or 0.5%, 0.5%, basically. And I went through and got the entire blasted area. And I also went out and looked at the surrounding genes. And the interesting thing about protocadherins is they're found in a big bundle on chromosome 5 in humans and chromosome 4 in chimps. And they're all together, and they get turned on in this weird cassette timing manner to promote development of the human brain. So anyway, so there's that. And let's see, where am I going to go next? I think I want to go here, there, and then I want to go to wrong. Oh, okay. So I went out and asked ChatGPT, why isn't it there? And ChatGPT says that PCDH1 exists in the chimp with a note, annotation information not in current annotation release. So the gene is actually in the chimp, but it's not in the annotation. We haven't written it down yet. We haven't done the work to point the gene out. So that's one of the genes, one of the PCDH genes that we found, that I found are different, right? So I went out and did a lot of work and found out a lot about protocadherins and where they are, and found out that, yeah, that gene is really out there. So anyway, so that's one thing. And then what else did we find? There's my list, and there's my picture of my genes. And let's see, what makes protocadherins? Oh, okay, we don't want to talk. Protocadherins are things that stick cells together. Protocadherins are a different subfamily in that gene class or gene families, and they're very, very interesting because they they exist like a cassette, basically. Okay, this is my blast. We were talking about the blast. So let's see if I can see it here. Okay, so this is my blast area right here. Can you see my cursor? That is 57,925 nucleotides, right? And I found it in the chimp. With the red means that it's in there. And I showed you the text where it tells you how similar it is. But lo and behold, look where it is. All these PCD genes. It's right in the cassette that does brain development. Excuse me a minute here. Does that make sense, Scott?
Yes, it does.
Ask a question. Okay. So anyway, it's right there in that nest, and somebody just has to annotate it, and it'll end up being another gene that we show right there. I'm choking. Okay, these are cadherins. This is the cadherin family, and cadherins stick through the cell membrane, and they have something on the inside of the cell, which is right here. And then on the outside of the cell, they have these little hooks, these ECMs, I believe they are. And when you add calcium to your body, these things hook up like this. So they hook together. So you have one cell here and you have one cell here, and these hook up. And these have a hell of a lot to do with how organisms develop. And if you change the timing of which of those cadherins, the other thing about cadherins is they like to hook up with themselves. They like to stay within their own species. So they'll frequently go out. So you can selectively create groups of cells that will stick together with things that are like them, right? So that's kind of how your organs develop and how your body develops, basically. And this shows how the cadherins hook up. They line up with you add a little calcium, some calcium ions, and these things stick to each other, and that's why you're not flowing all over the place like a pool of warm jello. So anyway,
who says I'm not?
Asked ChatGPT to make him a better writer, and they gave him alcoholism.
Well, there you go. It's like a freaking genie, that ChatGPT.
Yeah, it is. Anyway,
or maybe like a cookie small.
Yeah. What else I got here? Oh, yeah, there's just a picture of a little bit about protocadherins, and that's a little sciencey part that I wanted to do. And there's my list. You see there's 3 of those PCD, and I'm pretty sure I would find the same thing for all 3. And in fact, if I went through this entire list and blasted them all, if I had 8 hours to spend doing this, I'm going to get my software in place so that I can do that automatically. But I'm pretty sure I would find that all of these so-called orphans are actually in the chimp genome. They're just annotated different, or perhaps they're a gene that is downregulated and doesn't turn on, and it ends up being a pseudo gene in the chimp and a real gene in the human. There might be one of these out of the 23 I found. So anyway, that's the whole story. So there is no, the thing is that if one of these genes was in the human turned on, but in the chimp, it never got transcribed, right? That would mean it was an orphan gene. We call it an orphan gene. But the nucleotide sequence is there in the chimp. It didn't just pop out of nowhere in human beings. So their criticism of orphan genes is that they think they just appeared out of nowhere, just ended up working. And no, that's not the case. That is what an orphan gene is. I challenge these people to find one orphan gene that has absolutely no homologue in any other primate and mammal. That would be interesting. Agatus is here. Hi, Agatus.
You know, they're treating papers like Christians treat scripture. It says here in this verse. I mean, it says here in
this verse, right? Yeah.
And therefore, no,
yeah.
There's no plenary inspiration. It's not inerrant. Go to the source and look it up, and you will understand if you are seeking understanding.
Yeah. And it's, it's the same thing with reading Genesis. It's like, oh, it says in Genesis. No, this is your interpretation of what it says.
Right.
So your interpretation, it's your opinion of what it says. It isn't some gospel, that they're dealing with. And what else did I have tons of bookmarks on that. I got a whole bunch of stuff that he went through, a bunch of other fallacies he came up with for the brain. Brain complexity, a fatal blow to evolution. And we could get into some of that, but I don't know. Let's see, what else do I have to show you? Oh, I can show you these. Okay, this is my gene map human sort numbers. Basically, these are the PCDH genes, and these are alphabetically sorted, so it's really not a great example. But these are the genes in humans, and this would be over here are the ones in chimps. And I have a list, and there are 40,000 in humans, 30,000 in chimps, I think, or I got that wrong. Yeah, 31,238 that we've annotated in the chimp. And you see all those locs? Those are likely to correspond to some named gene in the humans. But this tells you where they start in the genome, where they end. This goes all the way back to the gene to the left of it and the gene to the right, and gives you a gap. And I can pull those entire regions, show you how many viruses we have and all that stuff. So that's the software I'm working on. Working very hard on that. And what else we got here? I don't know what else. Pretty much killed the son of a bitch, didn't we?
Yeah, they're just really dying on the wrong hill.
Yeah, I think so.
They should have just said, "Oh, we made a mistake," or maybe no, they didn't. They didn't read the wrong paper. They just read the paper wrong.
Yep.
Or they read somebody who read the paper wrong. See, that's the thing. The author did say novel genes, even though in context it doesn't mean the same thing as orphan genes. So they're just not admitting they misread it. That sometimes novel gene just means a new gene you add to a study, but that's not even what the source said. So go with the source.
Yep. And let's see here. This guy goes on for an hour and a half with some bullshit. And let's see the last part that he said. Go to that. Is that the last part? Is it going to that? Oh, it's beach balling on me. You beach ball. What? What's up with that? I got a lot of stuff open on this computer. Maybe I should shut some down. Don't know what's going on. Damn. Shit not working. Oh. Huh. Well, that's too bad. Anyway, what's going on, guys? Resist oppression. What's going on in here? We're showing creationists why they shouldn't use this goddamn meme in the future, because it's been completely shattered. And we're hoping that they will shut the fuck up about this nonsense.
Can you pull up Rebekah's comment to you where she talked about not wanting to spread misinformation?
Yeah, let's see here. That would be, nope, that would be here. That would be one more. Why is this not cooperating with me? Let's see. It's just not cooperating at all.
Well.
Well, hang on a second.
I was just going to say that the irony, it's just all over. It's just tingling all over my tongue. The taste of iron, and the irony of her saying she doesn't want to spread misinformation because that's all she does. She and her guests,
I know.
They just spread the most bogus crap constantly. And
here it is. I'll show you in just a minute. Let's see. Okay, there it is. Hi, Mike. Thanks for your substantive comment. If there's a serious misinformation in the video, I definitely want to correct it.
And of course, is she okay with minor misinformation? I don't know.
Maybe, I don't know. Maybe
serious misinformation? No. Yeah. Well, you're the lady who gives Cornelius Hunter, one of the most ridiculous anti-evolutionists out there, a platform. So, really?
Yeah, I know. And this guy is even worse. This what is his name? I keep forgetting his name. Uditha something or another. Anyway, he's horrible. I mean, he just says like the dumbest shit. He's just like, oh, the brain's so complex, and he goes on and tells you all the things the brain is hooked up to and all the things the brain does. How did that evolve? How could that have happened with random mutation? Just on and on and on. Anyway, so to sum up, the papers did not say anything like what he got out of it. And he is basically, if he says it again after we did this, he is lying to you. And he's probably going to lie for Jesus, because I guess that's okay. Don't they get a pass when they go to heaven if they lie for Jesus? If they lie to, bring you to the faith? I don't know. Anyway. So there we have it. What else? Oh, I was gonna try that video again. And for some reason, yeah, it's still beach balling on me. Let me see if I can go somewhere else.
By chance.
Oh, here we go. Okay, why is it beach balling at the very end? I don't get it.
At the end of a video? At the end of where?
At the end of the video. God damn it, stop it.
Maybe it recorded them trying to close out from StreamYard or something.
Okay, I'm going to put this back up. Let's see if I go there and then I do this. There we go. Let's see what he says at the end.
A theory which originated because of a change in the shape of a beak.
Yeah, I have some other questions to ask you, but I wanted to mention your book, Intelligent Design as a Proof of Creation. This is Dr. Jayatunga's book. It's very accessible if you want an overview of, the problems with evolution, the evidence for creation. This is really a good for people who are getting into this and would like.
Yeah, his book is not available.
In fact, I believed in evolution in my young days, but I always had doubts. And as I through medical school and sort of when you understand science more and more, it simply doesn't add because our biological systems are so complex. It cannot be based on 2 or 3 mechanisms as evolution trying to describe. Our systems need hundreds or thousands of mechanisms, right? I'll just give another example, Rebekah. If I want to buy a new house, right, I will look at the neighborhood, the area, the the road network, whether it is liable to floods, crime rate, employment opportunities, hospitals. Now, when there are multiple factors involved, before deciding to buy the house, only an intelligence or computer system can decide what is the best option. So when there are multiple factors are needed in making a decision.
Okay. Anyway, enough of this shit. What do you think? Oh,
well, I'm typing it out right now, so I was muted. But I wonder how many of the thousands of peer-reviewed neuroscience papers covering the evolution of the brain this guy has bothered to read.
Yeah. Yep. Yeah. Oh, there was one more thing I kind of wanted to show, and that is.
Well, go ahead.
Go ahead. So basically, so he asked me for my sources, and he asked me for a methodology, and this is basically I start with these massive 10 gigabyte files, and they have all the genes in them, and it's called it's a GFF, and then I break them into chromosomes. So I have like the C gene 12 is the 12th chromosome for the chimp, and then I have to let's see, I have to go ahead and select out, because there are hundreds of times more items than what is considered the best ref-seq, is what they call it. So I went through and I found the best references, and if that wasn't available, I'd go down to the next best, the next best, next best. And I took all the genes and I came up with this selected genes. And then I came up with, let's see, I had to clean them because they have all the columns, like they have 20, 30 columns crammed into one column. So you can't read it. There's no way to read it. So I had to flatten it and take it out. And then I went ahead and from that, I picked all of the genes for cell. And I picked all of the other things like enhancers and pseudogenes and shit like that. I went through and got all the signs, lines, and repeats, and that's a whole nother system that I haven't documented yet. And then I went ahead and I created these dir files, which is what I used to do this comparison. And these dir files, I concatenate them all together for all the chromosomes for the chimp and the human, and then I sort them, and that gives me the base information to start with to find the gene in the human or whatever. But anyway, there's a whole bunch of shit that's been done here that I have to document a little better. I think I'm not sure if I have.
Looks like you're well on your way to doing some pretty good documentation there.
Yep, I think it's pretty cool. Anyway, so I try. Wow, what happened to my camera? My camera's fading. Anyway, okay, let's see, brain scans. Let's see, what do we got in chat here? The field of medicine that guy went into was rehab, rehabilitation for walking and gait and things like that. Let's see, human program computer needed. Okay, what field of medicine? What else we got? He probably only goes over case reports for work. And a nuclear tidal wave. I help neuroscientists publish their research. Yep, for my day job. And we know you very well. They don't read research papers unless it's clinical trials or case reports. Has to do with surgical reports or to see if a drug works, but they don't review molecular biology research papers. There's no point. And brain scans. Okay, yeah, physical therapy is what he does. Basically, he's a doctor of physical therapy, so I think he's a little more than just a physical therapist. But he doesn't know shit about biology, and it's pretty obvious listening to him. He knows a little bit about the brain, it seems, but anyway. Yeah, I'm getting younger, Spooky. I'm reverting because I'm old enough now. I'm going back into my childhood state. Pretty soon I'll be drooling on myself and, you know, slamming food in my eye. That's the next phase of this thing. So anyway, okay, guys, I'm done.
Well, we came in just under an hour. That's pretty good.
Yeah, and not only that, but in 3 hours we're going to be on SNATA on Dave's channel, and we got to rest up because last night I was up doing this stuff in the middle of the night. Got about 3 hours of sleep, so I got to go take a nap.
Now, I'm going to go ahead and put that link to Dr. Joel Duff's video on preserved tissue in Devonian fish, because he's doing it as a way to get ahead of creationists saying, look, they found preserved tissue in a fossil. Ha ha. The Earth is only 6,000 years old. And so he went ahead and did a video on it. And so that way people can knowledgeify themselves.
Did you post it?
Did I what?
Did you give me a link to it, or did you put a link out in chat?
It's in chat.
You did. You did earlier, but it's
I did earlier, and I just put it out there again.
Oh, you just put it out there again. Okay. So I wonder if I can actually, yeah, you see, can you give it to me in private chat? Because that thing truncates it. It's got that ... and that ain't going to work.
Truncations.
And I'll put it in the video description. So the video description, I've got the 2 papers. I've got a link to the video that we're picking on, and where the supplementary data is for the Savage paper. And anybody's welcome to go ahead and tell me why I'm wrong there. I'd love to know.
Yeah, and of course the guy's whole argument is an argument from incredulity.
Yeah.
The brain's so complex, how did it evolve? Well, read papers on brain evolution.
I'd really appreciate it if you showed me where I'm wrong, because I've only been wrong once before in my life. And that was when I got married the second time. But other than that, you know, I'd like to be wrong twice at least. It's on my bucket list to be wrong again about something. So if you can point it out, I'd really appreciate it. Thank you. Thank you for your attention to this matter. Okay, Scott, we need a nap.
Yeah, you know, I do. I tried to kind of lay down. I got up early because of the dog and couldn't get back to sleep. And I tried to take a nap and I kept getting interrupted. So I probably do need a nap. Just, you know,
we do
a power nap. Let's not sound like tired old men. It's a power nap to increase performance and productivity.
I'm going to set my alarm for 4 p.m. so I'll be on a little bit late, but I will be on the stream for Santa anyway.
Hello, Ryan Murphy, just popping in with a bad joke.
Yep. Anyway, yeah, it was a mistake. Okay, guys.
See y'all later. Thanks for stopping by.
Thank you for checking this out, and I really hope I did a good job here. I did. I try anyway. Okay, guys. Goodbye, cruel world.